Hey guys, it's Mr. Wolf here, and I'm going to be going over the process of meiosis. So before we do this, let's just talk about the end goal, the purpose of this process, and that's to make these haploid cells, these gametes, these sex cells, which could either be sperm or egg cells. And not only do we want these cells to be haploid, we also want them to have genetic variation. That means that the cells that we get out of this, we want them to be completely genetically different to one another. We do not want them to be exactly the same like they are in mitosis. So how is this process going to go down? Well, we're going to start off with a diploid cell, just like any of the cells in your body, and we also have these homologous chromosomes right here, right? One from your mom, one from your dad. In this case, we have two sets of chromosomes, one short one and one kind of like medium-sized one in this case. Okay, so the first thing that's going to happen, and this also happens in mitosis as well, is the DNA is going to replicate. So here I'm going to have this chromosome make a copy, this chromosome make a copy, and let's have this one make a copy. Okay, so at this point, I have 1, 2, 3, 4 sets of the small chromosome. So this is not a diploid cell anymore. It's what we would call a 4-end cell. This is just only going to be temporary, but technically it does have 4 sets of the small and 4 sets of the big chromosome. Okay. So at this point during mitosis, the cell would split, everything would be genetically identical, but remember our goal is to have this genetic variation, so some things are going to happen at this point. Right? The first one is going to be called independent assortment. And so these chromosomes, I had them lined up before, but they're going to line up. Okay, so they might line up like this, but they don't have to line up just like this with all the yellow on this side and all the red on this side. They could have easily lined up like this. Right? And then when that happens, when they split, you're going to have different combinations each time these chromosomes line up. Right? So in this case, I'm just going to have them line up like this. Right? So this independent assortment is going to help create this genetic variation in this case. Okay, the next thing that happens is this thing called crossing over, or synapsis. So while the chromosomes are lining up, some of them are actually going to swap parts. So now you're really mixing things up.
Yell out.
All right. So now if we look at our cell, all right, every chromosome is now different from one another, even the ones that replicated, because we had this swapping, this crossing over. And again, the whole point was to create this genetic variation. Okay, so now what's going to happen is this cell, since we want to make a haploid cell and we now have 4 sets, not just 1 set of chromosomes, so we're going to need a split. So let's erase this cell here. These chromosomes are going to go this way, these chromosomes are going to go this way, and we're going to have 2 new cells. Okay. And at this point, we're back to our diploid cells, right? We have 2 sets of each chromosome, 2 sets of the small chromosomes, 2 sets of the mediums in each cell. And if we look at our cells, like they look very different from one another genetically, right? So we've accomplished the genetic variation, but we haven't gotten to this haploid point yet. So they're going to need to split one more time. So let's erase these cells right here, get the diploid out of the way. And now these chromosomes are going to move over here. These chromosomes are going to move over here. These are going to split. And what we're going to be left with are 4 new cells. And if we look at them, they are haploid now, right? I only have 1 set of the small one set of the medium in each cell. Not only that, but each cell is completely genetically different than the other. So I've created this genetic variation in my cells. So these cells could be sperm cells, or they could also be egg cells as well. And just a short note is that with egg cells, you create 4 of them, but only like 1 ends up becoming the egg cell. But overall, we've accomplished our process of meiosis, making our haploid gamete cells that all have genetic variation.